talks now framing the problem. And in general, it's meant to help the spatial program to frame questions moving forward, priorities, to define first projects, minimal viable products, really to bring technology into Atlas building projects. And we are here as a track really looking at it, technology and technology landscapes, but then also I would love to hear your opinion where you are within your lab, within your companies, building atlases or using spatial technologies in this context. And we have a few framing questions prepared. Can you bring them to the screen?
So in the meantime, I think we'll maybe also take the opportunity to present ourselves and kind of the type of work we do. So I'm Sanja. Most of what my lab does and what I've been doing the past 10+ years is very, very early technology development. So I was part of the spatial transcriptomics effort that became Visium and later on high definition spatial transcriptomics. So I kind of work a step before Holger. Holger will always be the one that will tell me, you know, this is working, this is not working, we should think about this standard or how to apply it. So I think we both need to hear from the whole community about where you're at and what next steps we need to think of as well in concordance with you. So Holger, maybe you want to present a little bit about 2 sentences about your lab.
Yeah, so I'm Holger Heyn. I'm leading a back then single-cell genomics group at the Spanish National Center for Genomic Analysis. Now we are moving obviously into spatial, having kind of seriously invested the last 3 years to build like a portfolio of spatial technology. So we're not developing spatial tech, but we're using spatial tech mainly in the areas of building atlases. That is one, but also in the areas of clinical trials related to immuno-oncology, really bringing back then single-cell tech and now spatial tech into the clinical context and to see how those technologies can be used in clinical trials to inform about drug action in vivo, and for us to bring those technologies from the academic context closer to the pharma context or closer to the clinics in the end. So again, we have seen a lot of technologies, tested a lot of technologies over the last years, starting with single cell and now with spatial. And as a community, I think we decided technology is ready to be applied at scale. And that's maybe one of the first questions I have for you, if you have the same opinion, because in the end, as in Atlas building projects, we are working now to build a roadmap and to invest seriously into spatial tech being applied to build atlases. And to my opinion, this could not have been done a couple of years back. Maybe we are already too late, so happy to hear your opinion. If the tech is already ready to build atlases, or are we already too late? And maybe that's a question for you first, because you're developing tech, so you're very early in the process.
Yeah. So traditionally, you know, we could have released ST, you know, it was published 2016. We had 2013, 2012 data, but, you know, it was one experiment worked, 25 did not. So, you know, my philosophy is we need to, as you say, build the minimum viable product, but that's my opinion, and that's how I run my lab academically. I don't think it's every tech developer's dream to, you know, work another 3 years to make it the minimal viable product that can be used outside of their lab. But that's, you know, we, as a community, then stress test each of these technologies, right? If I can't set it up in a month in my lab and my lab does tech, then probably no one else can set it up ever. Are we there? Yes. Yes. I think we were there even 2 years ago. But you know, there still is a lot of things we're learning from the single cell data, so I don't think we should just discard that. I think labs started integrating spatial and single cell data 2 years ago, and I think that stress tested the technologies even further. But today, I think the point of the discussion is to figure out which technology can we apply where and why. So maybe that's a question for you, because you're the one that's been applying these technologies everywhere.
But what do you think was brought us here? So what makes the transition from technology developed in a lab to being applied at scale in a community? So it's always commercialization, or is it also kind of ease of use, maybe also in the customized academic? So looking at NovaST, OpenST now,
Yeah.
with the newer kind of high resolution, very cost-efficient methods coming up,
Yeah.
as compared to commercialized methods that we know work probably at scale, so they're more established. So is that
Well, my counter argument is that they took a concept that already worked very well, but made it more open source. So we're maybe at that stage also in spatial technologies, is that we have optimized and re-optimized and reinvented some of these things. We know where to push them, and we could, or we can, or they can as well, make it more open source. So I think that's a big plus.
good? I mean, maybe a question for everyone, because I mean, now we have spatial genomics methods more established. So what is it you're using in your labs, in your companies? So you have methods, maybe we just make a quick poll. You have methods established in your labs? Yes, Mike. And that, or you can go through it, that's a Xenium system or a cosmic system, Merscope. So it's kind of a split landscape. And what's your experience? So you're happy with systems? It's you unbox and it's ready to go, or it's more a challenge implementation? Not even talking about the analysis side, but obviously from the tech. Do we need a mic? Yes, no?
We have one, yeah. So we need the mic because of the online, the Zoom stream.
So in my lab, we have a strong interest on mucosal biology. So we've been working particularly on the intestinal mucosa. And we started trying to apply vision when it was still based on oligo-DT, and we failed for quite some time because we realized RNA degrades quite quickly, particularly in the epithelial layer. Because we imagine that this layer of the tissue is trying to fight against this huge viral load that is in there. So that's something that we noticed. So we came up with RNA rescue spatial transcriptomics with Joachim Lundberg. So that was probe-based, that helped. Later on, we tried to apply Merscope, but then we failed again because of RNA degradation and perhaps lack of signal amplification. And that was somehow resolved by Xenium in situ. So in general, nowadays for the tissue we're working on, we prioritize vision, but probe-based, and then Xenium in situ because of the padlock amplification. So that's my global view on spatial technologies applied specifically in the intestinal mucosa.
So you think methods would fail more? So even if the kind of technology is robustly implemented and kind of scaled, it's still the tissue optimization where for challenging tissues, an experiment would fail.
Yes, ideally, I think there are some efforts on that direction, but they are not that well established, perhaps. Ways of actually preserving the RNA quality in those samples, because we are working in a hospital, we get our biopsies 5 minutes after they are collected, snap frozen directly, but we actually process the same biopsies across time, and you see that even under optimal storage conditions, the RNA quality would degrade. So I think we still need to figure out ways of preserving RNA quality in order to explore these tissues with broader applications that are out there. I see that Vision is now thinking about, 10x Genomics proposing to have a Vision ATD based on 3'. For instance, that is an application that I will never consider in the current context. But I also see that Vision is now improving Merscope with the Merscope 2.0 with a higher amplification of the signal. So perhaps in the future I'll be able to use Merscope, which was not since then. So yeah, I think this is kind of the horizon that I see out there.
Thank you.
Maybe someone else that wants to share their story about their tissue to just stimulate the discussion a little bit.
The mic run.
Yeah, I can also add something about the comparison of Xenium versus Merscope. So I work at a core facility where I'm responsible of the imaging-based spatial part. And so we've tried both technologies side by side over the past year. And indeed, I agree with the previous speaker that with Xenium, we had less problems to optimize the sample flow than for the Merscope, especially for two reasons. Indeed, if the tissue is rather degraded in terms of RNA, it's easier to still get signal with the Xenium because they amplify the signal. And second, also because of the autofluorescence. So from one tissue to the other, this can be very different. And that's much more relevant in the sample prep for the Merscope rather than for the Xenium, because in the Xenium, you just amplify your signal so much that you're not bothered anymore by autofluorescence. So indeed, we saw a huge difference in the amount of time that you spend on optimization in Xenium versus Merscope. On the other hand, for some projects, once we optimized and once we were able to run Merscope experiments, they also really had advantages. The specificity was much higher. It's easier to deal with samples that have GFP probes, for example. Customization of the probes, they were also earlier with this. So pros and cons on both sides is what we've been seeing so far.
Thank you. And maybe Christoph, come. Just also to learn about your decision-making process when you decide to buy one or the other technology. And I will ask you later, Christoph.
Hi, I'm Juveria. I'm from India. So the challenge that our lab faced was very different from what we are discussing right now. So India is an LMIC, we don't have a lot of money. So I have worked with Vision version 1. So what we did was we wanted to analyze multiple samples, but we did not have those many slides, right? So we integrated tissue microarray with spatial transcriptomics and generated the first data set. And then the whole technology can be independently replicated in individual labs. So, and the core size and everything, multiple cores can be set. So our challenge was to analyze how the different tissues are expanding when you float them in water, or how the tissue placement is done and everything. So, so I would like to pose one question, since we are moving towards developing spatial atlas. I wanted to specifically ask about, submitting spatial data. Is there a regulation on the minimum size of the tissue that is accepted? Because since we are working with microarray, many times there is a specific region that is pathologically very interesting. Not all the regions are. And sometimes the pathologically interesting areas are located far off from each other. So we take out cores and we analyze, and using that method, we have developed deep phenotyping of a specific lung cancer. So we have taken different normal edges into the tissue, different cancer regions. And there's a poster that I'm presenting today on that. So what are your thoughts on that? Yeah. Thanks.
Yeah, I think there won't be a hard cutoff of acceptable area or scanning or analysis size. I mean, we've seen the same from the single cell side, that in the beginning it had been 100 or 1,000 cells. Now it's 100,000 or 1,000,000 cells. So it's scaling over time. And for spatial, it's still early days. So I don't think there will be kind of stringent cutoffs how to contribute to building an atlas. In the end, it's more computational challenge in the end to stitch if the areas are smaller. But it's what we can do today with the budget we have in hand and technology that can kind of scale kind of across larger area sizes.
I will kind of summarize a little bit and say we have talked about two things and started two topics. One is, as far as I can interpret, how do we know which tissue is good enough to process with which technology? So a little bit about the QCing and collecting some of that data across technologies. And the second one is, well, you know, we're going to have a lot of different technologies with a lot of different tissue sizes. Can we actually integrate them? Will we integrate them? I don't know. That's a hard question, the second one. But
Do we have computational people in the room?
well, yeah. But maybe also to go back to the first part, right? Every vendor, every spatial technology has their own QC that they recommend to do with the tissue. What are people's experiences with that level of QC? Is that actually successful? Can you rely on the DV200 or can you rely histology plus RIN value? How do you decide, okay, this sample I'm actually gonna process with the Xenium, Visium HD, a MERScope? Because I don't work in a core, so I work with very specific biological questions. Yes, gut mucosa is one of them, but there are many people in the room that work with a lot of other tissues that might want to pitch in how they QC their things. I'm posing this because we need to know if we are going to do some QC and benchmarking as part of the spatial effort, similar to as was done in the single cell effort, and Holger did in his presentation also stress that we're not sure how to do this. And if we need to do this. So that's why I'm kind of opening the discussion to that, and definitely coming back to the minimum viable tissue size and product we want to put in the atlas.
So any QC experience from core facilities, maybe?
Again, better you than me. I work with not that many different tissues.
So indeed, for the MERScope, they recommend a certain DV200 value below which you should not go. We tried a bit below, and indeed the recommendation is quite accurate. Yeah, so at least that makes sense. But yeah, indeed, heavily degraded tissues, old tissues, tissues that were extracted in a way that was not keeping in mind to keep the RNAs viable, right? But that were taken with the patient in mind. Indeed, we see that the quality is much lower, and that then it's probably not the best technology for that specific tissue. Same for autofluorescence. This is something you could test in another type of imaging experiment, right? To see if your tissue has a particularly high amount of autofluorescence or not. Any kind of cheap IF or simple RNA fish experiment would suffice to see this. And there's also lists where you can check actually, I mean, it's rather known which tissues have a lot and which have less autofluorescence. So based on this kind of information, you can choose one technology or the other, I would say. On the other hand, for the Xenium, they don't really give a cutoff value in terms of DV200. I have not yet experienced tissues that are really so low in quality that were not giving a good run, but yeah, I'm not saying they are not.
Okay. Ellen has someone from online.
Yeah, there's a question from Anna Wilbrick-Clark. Has anyone seen any correlation between RNA-scope and success in Xenium and others?
Okay, so just to repeat, because your mic wasn't on, is there any correlation with an RNA-scope experiment and a Xenium experiment? Has anyone tried to do this? Both are amplification-based methods, so just less multiplex than one or the other, right? Well, we do a lot of RNA-scope, sometimes just because we're interested in one or two targets. In our hands, it works well. It's a kit that's been out there forever. We haven't tried in any other way optimizing it. So it's just kind of like, I'm interested in 2 to 3 targets. I'm just going to go to RNA-scope. I'm not going to try to design my in-house probe, order them from Stellaris, and do things when I know that the RNA-scope kit will work just as fine, even if I order some probe synthesis from them directly. Whether we have done a direct comparison on FFPE between RNA-scope and Xenium, not really. Just because we don't handle that type of samples or questions from an academic standpoint. On fresh frozen, we have done some. We have also done different types of in situ sequencing. Xenium-like, hybridization in situ sequencing versus actually sequencing bases. Yes, there are huge differences in biases in that chemistry. One is better for some probes than for others. So I can also say that my lab or one lab cannot test all these probes. We have to also say that none of these probe panels will ever be perfect. So we just have to mark the ones which are not. Okay, that's kind of my 2 cents on this. We're never gonna have 18,000 probes that don't bind to other regions.
Yeah, wait.
This is Mike from Tex. Just on that question, we haven't done that internally, but I did speak to a customer recently who is trying to do that, so I can get the names of anyone who's curious about that, and I just need to ask if that person's willing to talk to you, but they haven't done that analysis yet, but they are trying to look whether there's a correlation there. I'll just point out, because we've been trying to do some of this ourselves too, one of the challenges is like the end that you have to get to convince yourself that there's a really good correlation is pretty tricky. So, but anyway, if you feel free to, I can give you my email and they can reach out, and anyone else who wants to.
Yeah, I think that would be true for any type of fish versus sequencing-based methods in space. So we've done a lot of these experiments, and usually you end up having an N of, 8 mice and 40 tissue sections in 2 technologies to do that. Yeah, that's expensive for any lab. I think what we have made progress with is making recommendations on the kind of power studies that you need in either of these technologies to be able to answer some of the questions. So I'm also counting on, Mike's team or Bertrand's team or someone else's team that has more resources on the industry side to also give out some of the recommendations between these technologies and where do they fail and scale.
Okay, so given the quality of a sample is good, so how do you kind of make the decision, just touching on these points here, of using an imaging-based, a sequencing-based method, if imaging a low-plex, a high-plex? So that's mainly based on accessibility, budget. So what's your decision-making process? And also when buying a device, so how do you decide between vendors and technologies out there? I can start with our experience. So we've been working basically on all systems, mainly motivated by the panel sizes. So we come from the sequencing side, single cell sequencing. We're a sequencing center in the end. And then working on single cells, we have been initially focusing very much on the capture-based methods, because in the end it's random capture and resolution increased over time. But especially when panel sizes increased from 500 to 1,000, now multiple thousands, that's where we really became interested also to apply those technologies at scale for atlasing type projects, eventually with a goal to replace cell dissociation and move entirely to imaging-based atlasing projects. So that's a goal. There are a lot of challenges still. One is the panel size still, although with the 5,000 or 6,000, we are pretty good in representing the transcriptome. But then there are challenges like cell segmentation, where we are still not sure if what we are measuring or what we end up in the analysis is really a single cell representation, or still contains a lot of artifacts. So that's where we are coming from, but happy to hear kind of what's your decision-making process when deciding on a tech or technology application. Yeah.
So we have also a Visium, Xenium, single, basically we have everything, I think. So the sequencing versus imaging, I think usually the first question is, do you really need single cell information? Because in all the capture-based technologies, you will never identify individual cells, right? You get areas, and even if the resolution is small, that still never represents a single cell, right? So if people are interested in rare cell types or cell types that are isolated from others, and if it's about cell-cell interactions, all of these kind of things, it's really cells that you have to see, right, and not capture areas. So in those cases, we usually recommend imaging-based technologies. On the other hand, it's a targeted technology, so if people don't have a good idea yet about the genes to look at, we often recommend to first do simple single-cell experiments to have an idea about the genes that are relevant. Doesn't have to be a spatial one, just single-cell experiment to get an idea, to be able to make your list of few hundred genes that you would then look at spatially with imaging-based technology. Then on the other side, if you're interested in full transcriptomic and you don't really need to see individual cells, but rather areas of the tissue, then a VZM experiment is very interesting because you directly get full transcriptomic and spatial information all at once if you don't need to identify really individual cells there. And
Oh, okay. I would counter-argue that with the Visium HD, you have the 2-micron resolution and you can bin it in a cell.
but you don't know where the cell is, so it's the size of a cell, but it's not a cell, right?
Okay, yeah, but
it's not the same thing.
sure. I'm just saying that there are other staining methods you could use to do
I
a cytoplasm.
think you definitely, if at some point you could overlap a staining image where you have individual cells with a Visium HD experiment and you could bin all your tiny capture areas inside one cell, then it would be really interesting, but that's not there yet in that way to really combine an imaging and a Visium experiment in that way that you can really identify cells and then have full transcriptomic information on those cells.
Okay, and can I ask if you have, by any chance, evaluated, on the Xenium, there's, as far as I remember, a couple of versions of the kit. Some of them I might already now have introduced a cytoplasmic stain. Do you think, in your hands, have you tried a little bit of data analysis-wise segmentation of that type of data?
Yeah. For the staining, it really is really tissue dependent if it works well, because it's a general few marks, right, that they chose to outline the membrane, the cytoplasm, the nucleus, and for some cells, some tissues, that's really appropriate stainings, but for others not. So I've seen cases where it works super well, and I've seen cases where it's terrible and people need another method to segment.
Okay. I think we touched most of the points. So moving to the next question, because we talked about the landscape and how we choose technologies, challenges, preparing samples. So what do you think is missing today? So moving maybe not only into Atlas and Atlas generation. So what do we give 10x other companies as homework to be developed to address challenges, to be better in building atlases, but also, for example, functional screens? So is there anything you have in your mind to put on a list?
Which kind of I'm always wondering which kind of resolution are we aiming at, because if you analyze at a subcellular level, you can also use the atlas to infer function of let's say different RNAs, different complexes that you may have in the cell. Of course, very high resolution is expensive also in terms of data storage, but yeah, I think is to me still an open question, because we can infer much more biology once we increase the resolution.
I agree. I will also add to that we would need to measure the different molecules. So, we would need to probably polyadenylate everything that's in the cell to capture it on a poly-DT array, right? So circular products, links, microRNAs, and so on. Yeah, would love to do that. In our hands, kind of storage-wise, computation-wise, sequencing-wise, we stop at 1 micron because, again, the types of questions we are after, we don't need more than 1 micron. And I would also open a little bit the discussion to that particular question because, 1 micron is, let's say, a xenium-like experiment. 2 micron is an HD experiment. 10 micron is a Curio-seeker. Stereo-seek is 25 or 0.25 micron. The only thing I've ever done with that data was bin it into larger data because computationally, like we cannot deal with even a simple task like inferring cell types at 250 nanometers. So I know the computational people are in the other session, but in my lab, we always have to bin it. So whether I bin it in quadratic bins, right, 8 times 8, 4 times 4, whatever we call them, or I bin it into what I think is a cell, because otherwise there's too few genes that are detected in a little tiny 1 micron times 1 micron spot that on histology, if you present this 1 micron spot to anyone, no one will know what it is. So I don't know, Holger, maybe you have a different view.
So you say binning is necessary, so increasing resolution does
Well,
not make
I'll
sense based
also-
on sparsity per
Yeah, I'll
spot.
also argue that, the last talk from Grace posited a little bit a thoughtful question about integrating histology, right, and the foundation models. Well, the foundation models, they don't run on 1 micron spots. They run on large patches of tissue to actually be able to infer anything. So I would love to, see how much histology can help us in these tasks. But it's not going to be a 1 micron histology patch you actually try to classify.
Speaking of these very small spots, obviously what you're saying is you get very few counts out of any of these spots. How much is that, how much upside potential is there to make the assays more sensitive? Like, has anyone done the math how much RNA should be there in any one of these spots? And also how much variation of RNA density should be there? And what our current sensitivity, quantitative sensitivity is, and what is the realistic sensitivity that could be achieved with certain optimizations of existing methods?
Yes, to some extent. I think every of the spatial methods that has been published in the last years puts a number to their estimate of efficiency or sensitivity, comparing it to single molecule fish, not comparing it to some theoretical number of 10 picograms of RNA in a cell. So just to single molecule fish of that particular target. How people have picked these targets, they evaluate efficiency on, it's mostly that it's a sparse enough target to easily either segment or is in different parts of the tissue, so you can compare efficiency in different parts of the tissue, okay? I think, to be honest, 10 years ago, it started at 1% efficiency. Current claims, and not by my lab, by many other published methods in the last exactly 1 year, that efficiency is around 20% for sequencing-based spatial methods. I'm not necessarily sure it's equally as fair to compare, when you have an RCA product and a Rolani there to count those spots as individual molecules, but that has also been done, and they have benchmarked it at around 10 to 15%. I cannot say too much about how much more we can push the Rolanis, apart from having a little bit different sequencing chemistry, because we will always end up in this optical crowding problem of detecting the actual individual spots. What we are working on computationally is different regularization algorithms to try to improve image quality to better accurately call spots. I have not seen a single benchmarking paper that will tell me okay, these are the objects at different resolutions. Can you please tell me which of the algorithms for spot calling works the best? Yes, it's compared to some other method, but not to some kind of synthetic data set that's been created. So I think that's maybe the first thing to better estimate efficiency in in situ sequencing type methods. For sequencing-based things like Visium, Curio, or others, yes, there is a way to push the efficiency, but maybe what I put out in my lab that pushes the efficiency, I cannot necessarily know whether that will easily push the efficiency on an HD Visium. Okay, so we need to have an open dialogue with companies in the room to actually, me very early on, tell them, hey, we have found something. Are you guys actually interested? We think it will push it so much further. So it gets integrated into the kits. Because all of us do the R&D, right? So we just need to talk also a little bit more openly about it. Yes, there are ways to push the efficiency, but sequencing then, saturating the sequencing is a little bit different ballpark that we also should maybe talk about a little bit.
And then you think methods like slide tag can be part of the solution, because then it's a sequencing, single cell capture, molecule capture problem again, and efficiency might increase, not talking per cell, not talking about the gaps on the tissue, but kind of what is captured, transcriptome from a cell.
Well, with a few groups in China that have in the last 6 months released some very, very intriguing spatial tech that's not built off of anything we've done so far. And all of those have much higher efficiency in sequencing-based spatial technologies just because of the chemistries they use. And I'm happy someone did that for us and we don't have to. Let's put it that way. I have a very hard time, slide tags as a concept, just for people who are not familiar in the room. It's kind of taking the slide seek technology or the Curio seeker technology. You release probes into the tissue and then you collect that material into a 10x prep. So the droplet-based single cell preps. I don't know how to infer, for example, spatial information from that type of analysis when I'm sampling only 4% of my tissue. So I don't know if I build algorithms that are spatially aware. I don't know how to deal with this information that only 4% of all the cells that I'm sampling are actually sequenced. But I think we could, if we, okay, I'm speculating now. I haven't really thought about this. But maybe at some point the spatial atlases will be at a stage that we don't have to very densely sample tissue. And we can very sparsely sample tissue and it will be enough if we give enough guidelines what we're looking for. I don't know. Same kind of like we're thinking about, Mike as well presented at the 10x talk, that they are using the 5,000 panels in Xenium to actually better inform the smaller panels. Can we think of the same way in sampling spatially cells? Do we actually have to sample all of them? Because remember, in the single cell world, we're sampling, from a Gaussian distribution, a whole tissue, you're sampling from a Gaussian 1,000 cells or 10,000 cells from that little block. Can we imagine the same scenario in spatial data? Yeah. That's kind of what I'm thinking about the slide tags.
It's like dealing with sparsity, not per cell, but per tissue.
Yeah.
But maybe you can also comment where we are today in terms of how much, the slide tag technology, how much of a tissue is captured from using the original version slide tag that is published or what is commercialized now, and where are you going?
Yeah. So I think psychologically a lot of people hear like 10%, 50%, and they think that's not workable. But when you actually see the data, it's pretty impressive actually. But for an example, a whole mouse brain will recover about 100,000 to 200,000 nuclei from the tissue, which we would estimate to be about half of what was originally there. And of what we recover, if we sequence maybe, say, 30% of those or 40% of those, it actually looks very dense in terms of what you visually see. So I think the number of nuclei, you can scale the number of nuclei you want to sequence, right? Because you just put more nuclei through the single cell platform. So I definitely think it's higher than 4%.
Yeah, I think
Right.
that was the theoretical calculation. If I would sample from Gaussians,
Okay.
double Gaussians, it would be
Okay,
4%.
Yeah, so,
I think the original publication states 10, but it's like 5 to 10 doesn't matter. It's,
yeah, I think
know.
it as a commercial product now, it's a lot more robust. Right, yeah, so I think that percentage has definitely gone up since then.
Yeah. My point is just, it's not every cell that's sampled, so we have to think about what to do in the analysis steps, because currently we're treating that data set as ground truth, like this is spatial data, right? But we're not actually sampling every cell.
Yeah, and I think one of the major advantages is you're getting, because it is single nuke data, you're getting 2,000 or 3,000 genes on average per actual cell than you would get with other platforms.
And it's single nuke only, or it's single cell, or single nuke?
Single nuke. Yeah. Okay, I might, okay, oh, sorry, please.
Different topic. Does anyone do DNA or believe in DNA microscopy, like optics-free approaches? Is there any trajectory that this is going to be useful? Because this, you could totally see how you could do this in whole tissues very nicely and essentially have to give up any concept, or be able to give up any concept of what is a cell and just understand the tissue as it is. Has anyone worked with pixel gen type of stuff or cooked their own system? It seems the papers about DNA microscopy came out a couple of years ago, and not much has happened since, really.
So Josh is not here, so I don't want to speak in his name, but there has been 2 or 3 a little bit more recent preprints, one of which was from Josh's group at Chicago, where they in 3D profiled maybe a Drosophila embryo. I don't necessarily remember the details. I really like the concept of DNA microscopy. I think there's 2 caveats to it. One, you actually have to have proximity between cells for it to work, because you have to measure the context between the cells, or what Josh calls unique event identifiers, UEIs, I think. And that's not always, very easy to make work when cells are not very close to each other. And the second, I think another group, as well as Josh's group, so the other group is the Hoffaker lab at SciLife Lab, have for years been trying to scale computational approaches for these type of reconstructions. And based on their most recent work, and that work has been on bioRxiv the last 3 months, I would say, the reconstruction is not very accurate computationally. So there's always, call it, image distortion between what's actually, in your sample and how the distances you're reconstructing. I would love to say this is going to be scalable in long term. I don't necessarily know right now. But it's something that, Josh's lab, my lab, Faye's lab, other labs have been using and implementing. I just don't know, as a kind of technology person, whether we're 6 months or 6 years away from making this, in a real 3D scenario.
Okay, so we have around 10 minutes left. I think we touched a bit what's happening in the future. Maybe Orit, as a standards and technology co-chair, we've seen a lot of stuff happening in the last years. First single cell, now spatial. So where do you see we are going on the single cell spatial side, integrated? What do you think we're missing today? Yeah.
Well, I'll point out one thing that what my lab also works on is integrating imaging dynamics with sequencing. Kind of like when we think of the live cell, yes, we're doing it in vitro systems because my lab doesn't do organoids. We don't do much of animal work, so we're limited to 2D cultures. But I would also like to hear, we have heard from people that have dealt with difficult tissues with spatial data. Is there something, a certain, kind of, let's say, the core facilities would like to see today from these technologies? And then a more academic question is, should we be working on, 3D perturbation atlas in some model system? I don't know, right? But I need to hear from someone so I know what we need to work on.
Maybe again from the core facility perspective. So what's your kind of bottleneck generating data at scale? So it's the runtime of a Xenium, of a Cosmix, of a Merscope, or it's a prep time to really get there?
Or maybe something different, like you don't have enough communication with the experts, with the tissues, so that you just test the standard things. You've tested, for mouse brain, you're testing on the human colon, and then it doesn't work. Do you need more input from technology side, and do you need more input from the expert, let's say, networks or organ experts?
I think the cost of the spatial imaging technologies is so prohibitive that you cannot test things, right? So people usually, they get the grant, they do the specific few tissues, patients, whatever that they want. But then on the side of, where you can add a lot of genes that you do in one tissue and another one that you can compare, or that you can compare different optimization conditions, all of this, that's really something people just do not do because that's the money is really prohibitive. I think that's in the kind of things that you mentioned, really the bottleneck of any sort of, community-based baseline of how do we handle these samples, how do we compare from one lab to the other, how do we make protocols that we know that we're doing everything in the same and in the right way, because people do this one holy grail experiment and that's it, right? In my opinion, right now, I don't expect solutions coming from the companies, but we are on the side doing in-house fish also. So to test things in a lower plex way, but there at least you're fully in control. It's a cheap experiment. You can do things really the same across many different tissues, across many different labs, and you can do all your tests there. At least you will have some sort of comparability across. But right now, I really don't see that with the companies because I don't see any price lowerings. Actually, it's going up because then you need also your segmentation kit, you need everything. I mean, that doesn't seem to be the direction it's going, I would say.
Also, so a quality control kit, that's what we want.
Yeah.
Spatial quality control kit.
Yeah, that you can really do often on many different tissues in many different cases without being bothered by the price. I would say something like this, yeah.
Okay.
You have to get the mic.
I think we should really try to get this OpenST, NovaST to work at scale, because that's an assay where you can potentially afford a lot of individual experiments, just because every single bit of the NovaSeq flow cell, you can really break it apart into these tiny spots, and you can use a lot of these. There's so much value in an assay that you can scale, and where you can do tech development, tech optimization, etc. And since, like in theory, in the single cell field, we have these assays, like combinatorial indexing should be cheap, but it's priced based on competition rather than priced based on cost. So if there's now someone commercializing a very cheap assay, they would still charge the competition-based price, whereas with the Nova ST, etc., we have the advantage that the flow cells are being produced by Illumina for other purposes, and they are priced according to their use for other purposes. So hopefully that stays cheap. We just need to get it to work as a community, because that's an academic essay. So we need to do the tech development rather than relying on a commercial provider to do it. So I would encourage people to, when these open essays come out, to actually invest time, energy, money to set them up and share their experience, because then we have to do some of the tech development that otherwise the companies are doing, but in return we can get an essay where we can do a lot more experiments, a lot more samples than with the, as you say, cost prohibitive commercial essays.
But you think it's easy enough to set up for a lab, or that's a task for a core, or needs a company to commercialize and make it easier to be more accessible to the mass? Good.
May I comment? So many years ago, after the human genome, people realized that we should sequence many more genomes, and the cost was 300 million to make the first version of the genome. And somebody said, let's make it the $1,000 genome, and now is the $100 genome. So the companies may realize that we will have that grant, we will try to spend that amount of money. If you make it much more efficient, we may be able to have the data that may convince pathologists in the hospital to also develop that, and you may even maximize that market, perhaps. So we can have some, shall we have some discussion to make a call to having the $100 spatial transcriptomics, $10 spatial transcriptomics, because we will just produce more data, I think. We will not spend less. So I think probably we should have
So it goes through the economy of scale. So the more people use it, the more it's implemented into, for example, a diagnostic context.
They can produce at much lower cost, because in the future you can see that all of this will be used for in the hospital.
I think that was the initial idea when we made ST, is like one day it will be in a hospital, right?
Exactly.
Yeah, so I agree.
Shall the Human Atlas make this call, okay?
And when do we see that coming?
Not only me, but it's group.
I cannot comment too much about the pricing thing, because I know how much it costs to produce.
So then what is the limitation pushing it into the hospital context today? Is it proof of concept missing, or it's still too expensive per sample?
Honestly, when I came to Columbia, and the NYP Columbia system is like a whole world on its own. You know, when you have a chair of pathology that's 85, it doesn't want to change things, nothing will happen. But when there's a new chair of pathology that actually wants to see molecular data or whole genome data, now there's no problem. Now there's actually, you know, I don't have enough people to scale the workflows as much as I would like to. So yeah, I mean, some of us have the technologies in the hospital already, but of course, this is not for diagnostics use that we can dream of maybe one day, but today, no. I agree, this is possible because I can make those products myself. I support what Pierre is saying, and we should maybe make a call. Yeah.
Great. We're not wrapping up yet, but we should wrap up soon. So maybe just to pick up on the future of assays, switching to multimodalities. You've been working with microbiome data. I think there are many options out there today. So what's your experience, and if not experience, kind of need moving forward to add something onto your transcriptome profile? Again, a wish list or experience.
Well, I would argue it's just based on the question you want to ask, right? Ronk has demonstrated nicely some of these multimodal technologies like protein and attack and RNA, and so on, the combinations. We have worked a lot with histone modifications, DNA, proteins, microbiome, and so on. But when we went into the tech D part of those projects, we had a very specific question, biological question we wanted to answer in mind. that we are answering with the technology. But like, what more can we add?
Yeah.
What do you want? I mean,
But you would
this is
also
kind
argue
of
that
like-
for the bulk of the experiments and experimental designs, RNA is just sufficient. So if we have enough RNA information for phenotyping, immunophenotyping, do we need an additional layer, or it is just then a very specific niche to go into mechanisms, to go into function?
Well, if you would ask me, I would love to have chromatin plus protein data and skip the RNA, but have it at the same scale. That's probably me. I'll move. I would also argue that a little bit why you add protein sometimes or why you add chromatin sometimes is because the RNA signals is sparse, so you try to help yourself with these other modalities. That's not really answering much biology. Okay, so if we improve quality at all of these assays, I would say yes, multimodal, but not just to kind of patch things up.
Okay, I think we have to wrap up.
Okay.
Because there's a keynote coming up. Thank you all for being here with us, for the fruitful discussion, and see you in the main hall in 10 minutes, please.
Thank you, everyone. Thanks, Holger.
Thanks, Sanja.